the medullary cord, not by junction formation of the hinge points. Okay, we also saw how many neural tube defects can occur in the anterior or posterior axis of the individual, whether it is in the anterior portion, it will be called anencephaly, and whether it is in the posterior portion, we're going to have a spina bifida. Okay, that is the failure to close the posterior part of that neural tube. We have a differentiation of the neural tube in that anterior-posterior axis. In the anterior, we're going to have the ballooning of the vesicles. We start with 3 vesicles and we end up with 5 vesicles. Okay, and the cells along this anterior-posterior axis depend on different signaling centers. Okay, we have the BMP4 in the roof of the neural tube, and in the floor of the neural tube we have sonic hedgehog, which comes from the signaling from the notochord. Okay, and then we have the gradient between BMP and sonic hedgehog, sensory in the dorsal, motor in the ventral portion, and interneurons in the middle. Okay, so in this lecture, what we're going to be discussing regarding the neural tube is the derivatives and surface cells derivatives, how these cells generate. So the formation of the brain is one of the most important tasks of the vertebrate development. It's so hierarchically structured, and so many things can go wrong. So all animal depends on the well-organized and highly structured of the brain formation. Differentiation of the neural tube into various regions of the central nervous system occurs simultaneously at 3 different levels, and at the 3 levels that we have in the screen. One is microscopic, then we have tissue type differentiation, and then the cellular, because even though all of those are cells that belong to this system, the brain and the neural tube, they all have different functions. And the way how they're created is not necessarily the same. So if we look microscopically, so the gross anatomical scale, the neural tube bulges and constricts in different sections to form the chambers of the brain and the spinal cord. It's not just a tube, right? We saw the ballooning, and we have the constrictions that identify the telencephalon from the melencephalon, from the diencephalon, from the rhombencephalon. So you have constrictions. So that microscopic. So when we see a brain, a cut, we say, okay, this is the anterior, this is the telencephalon, here is the constriction, and then the next chamber is whatever it is. Okay. Then at the tissue level is the cell populations within the wall of the neural tube can rearrange themselves to form different functional regions of the brain and the cord. So like we have the myencephalon of the diencephalon, we have different elements in each of these sections that will form different structures of the brain. At the cellular level, the neuroepithelial cells themselves differentiate into various types of nerve cells that we call neurons, or supportive cells that we call the neuroglia, right? So they are way more neuroglia than they are neurons, because neurons require, these are high maintenance type of cells. So if we look at a brain of a vertebrate, they have about 170 billion cells between neurons and glial cells. That's a whole whack of cells, right? 170 billion cells. From those, we have some neural stem cells. Okay, so the first multipotent, and I just want to make the point, these cells are multipotent, so they kind of still create or become a selective number, but not all of the type of the cells, right? Multi is just less than totipotent. So can become a restricted type of body cells, and they make up the neural plate and early neural tube. So most of what we found in the neural plate section before the tube start forming are part of these neural stem cells. Okay. The neuroepithelial cells are highly proliferative and capable of generating progenitor cells for the first neural and glial cell types in the neural tube. These neuroepithelial cells will then transform into what we have here, the ventricular cells. The ventricular, pay attention because sometimes I'm going to call them the ventricular cells and sometimes I'm going to call them the ependymal cells. Okay, so ventricular is ependymal cells. And normally this one will remain part of the neural tube lining. Okay. Do you remember what is the function of the Ependyman cells? The ependymocytes? No, that says nothing. Those cells for the people that took anatomy and physiology. Okay, so the Ependyman cells are the ones that secrete the spinal fluid. Okay, so these ependymal cells, ventricular, are always in the lining of the neural tube. Okay, because they are the ones that are going to be secreting this spinal fluid that will circulate again and again every day to clean and to nourish all of the different cells of the nervous system. Then we have the other type of cells, and the cells are transformed into radial glial cells or simply radial cells. Okay, these cells will serve as the major neural stem cells during development, and they have this multipotent capacity and self-renewal. Okay, these radial cells, as we want to see through this course, they also help during the migration of other progenitor cells and the newborn neurons. Okay. We will discuss about the brain growth as we go, and we're going to see how these radial cells have so many polyvalent functions in the brain formation. Then we have neurons. This is what we relate a little bit more with the nervous system. So neurons are cells capable of conducting electric potentials and transforming that electrical signal into a specific function, even if a thought, a sensation, a perception, a feeling, etc. So that's what makes us what we are, right? And allow us to do what we do. So out of these 170 billion brain cells, the human brain consists to 10 to the 11th neurons associated with more than 10 to the 12th glial cells. So we have way more glial cells than we have neurons. Okay, we're going to have an anatomy of a neuron that by now I hope that being a biologist you know this basic structure of a neuron. So we want to have these branching extensions like sticks that pick up the electric signal impulses from other cells that we call the dendrites. Then we have the soma, okay, which is the body of the neuron. Some neurons may have few dendrites attached to the soma. Some others will have extensive dendritic trees, like the Purkinje cells, for instance. Like something like this. This is a Purkinje cell. This has huge, massive amount of dendrites. Okay. And then we have the axon, and then we have the growth cone of the axon. This is the axon helix. This is where the actual potential is generated to bring it along the axon and then do synapse with something else. This is a diagram from the 11th version because the 12th version doesn't have it. So all of this is the receptive part of the neuron. This is the transportation, and this is the end part of the neuron. Now, one important thing to to know is that during the first year of life of a human, there will be a dramatic increase in the number of neurons on the cortical region of the brain. Okay, so babies are not born necessarily with the amount of neurons that they're going to be the continuous. Normally, we say that babies are like living extrauterus fetuses, because normally this brain going to continue growing massively during the first 3 years of after born. Okay, if the brain was fully formed at the moment of birth of that kid, that brain, that head wouldn't pass through a vaginal canal. Every baby will be born by cesarean, believe me. Okay, there is no going down there. So the brain suffers a massive transformation during the first year of life of the baby. Those neurons will create as many as 100,000 synapses with all the neurons during that year of life, that first year of life. Okay, and hence will allow the cortex to act as a center of learning and reasoning. So that's why we see the babies start moving without coordination, and little by little they start having a movements a little more coordinated, and all of that is because they are creating new synapses all the time as they are growing. Another important aspect or feature of the neuron is the axon, right? So all of these axons here, this continuous extension of the neuron cell body, and maybe several feet. If we look at the ones that we have, for instance, in our legs, could be about 60 centimeters, 70 centimeters long. It all depends how long, how tall you are. Okay. Okay, so let's talk a little bit about these glial cells. So in the nervous system, we find 3 categories of glial cells. Okay. So we have oligodendrocytes, we have astroglia, and we have microglia. Okay. Axons normally are insulated with myelin sheaths, okay, to increase the rates of signal propagation and prevent dispersion of the signal. We know that normally a cell that is covered with the myelin sheaths has about 100 times faster the information from one end to the next. Okay. In the central nervous system, oligodendrocytes, glial cells, produce the myelin sheaths. Okay. So these are the ones I have here in green. These are oligodendrocytes surrounding multiple axons. Okay. The axons are the ones that are in purple. The green is the myelin sheaths in here. Okay. Now, in the peripheral nervous system, the cells are what we call the Schwann cells. Okay, so there are 2 different types. Central nervous system, the gliodendrocytes cover and produce the myelin sheaths, but in the peripheral nervous system, these are the glial cells that produce the myelin sheaths. Okay. So myelin is essential for proper nerve functioning. If a cell, if a neuron gets demyelinated, something like the multiple sclerosis, for instance, destroys the myelin sheaths, that individual may have convulsion, paralysis, and can have lethal conditions like the multiple sclerosis. Okay. Oh yeah, I forgot that. Just come on. 1 second. So in the axon, this is oligodendrocyte. So this is the central nervous system. You see how it starts moving and surrounding all around that axon. So we can have from 30 to 50 layers of this cell, just cytoplasm, just going and rolling in the axon. In here, we have a cell, it's in the peripheral nervous system, so it's a Schwann cell, and it does the same. Okay. Okay, the second type of cell of these glial cells are the astrocytes. We find radial glial cells and different type of astrocytes. We have type 1, we have type 2 that are reactive astrocytes. Astrocytes represent about 20 to 40% of the total glial cells that we're going to find in the nervous system. So it's huge. 20 to 40% being just astrocytes is important. These astrocytes establish the blood-brain barrier. We cannot allow everything in our body to go through, otherwise we will be so susceptible to viruses and bacteria messing up with the brain, and the brain is our center of control. So these astrocytes are the ones forming that blood-brain barrier, okay? They also support that homeostasis and neurotransmission. They respond to inflammation in the central nervous system and support the synapses for this. A condition that is called Alexander disease is a neurodegenerative disease caused by fibrous proteins aggregates that is present in the astrocytes as we see them here. Okay, so that of course will debilitate the transmission of the signal, but also maybe making the brain a little more susceptible for this barrier to allow the passage of many things that we don't want in there. The third and last type of glial cell that we mentioned is the microglia. And these cells are also known as the immune cells of the central nervous system. Okay, and they will phagocytose dysfunctional neurons and any other type of glial cell that is not functioning properly. And they can move through the nervous system, even though they are not born in the nervous system. They are rather born in the yolk sac of the embryo. So they go and migrate very big distances. And they're not too big, they're tiny, but they do a really important function in our body. Okay, so now that we know the different type of cells that we find in our brain and neural tube, now let's look a little bit on the architecture of the central nervous system first. Okay, so neurons in the brain are organized into something that we call layers or laminae. Okay, and also intranuclei. The nuclei are just clusters of cells. Okay, so either they are in layers or they are in groups. Okay, so either they are in laminae or they are in a nuclei. Okay, and they can have, and each can have different functions and connections. It all depends in the precise localization of these cells. So the original tube is composed by this germinal neuroepithelium, and normally is only one cell thick layer. Okay, and this is a layer of rapid dividing neural stem cells. Okay, so and it all starts like in this figure. In this section, we have the lumen of the neural tube, which means the center. Okay, and we're going to find different sections. The ventricular, which I told you that the ependymal section, right? So this ventricular zone is always next to the lumen of the neural tube. The second section is what we call the mantle zone or the intermediate zone. And then at the other end, we have the marginal zone, and is the outer zone of the neural tube. So the basic structure is this one: ventricular zone closer to the lumen, intermediate zone or mantle zone, and then the marginal zone, that is the outer section of the neural tube. Now, the genesis of these differential zones starts with the ventricular zone. So it all starts here. These are the ones that have these stem cells. They're going to start dividing, and then they're going to start migrating into the different sections. Okay, so they're going to start in the ventricular zone, and then they will start a process of migration. Going to have some sort of monorails that are going to tell them where to stop. Okay, and they're going to form the different layers until they get into the mantle and the marginal zone. Is this clear? Yeah, okay. So depending on the level at which we're talking about or discussing, the hierarchy will vary a little bit. So the genesis of these differentiated zones start with that ventricular zone, so the closer to the canal, to the neural tube, will continue migration of cells and layers formation, as we can see in here, until they form the mantle or the intermediate zone. Okay, so we have some stem cells, and we have the radial glial cells that will act, as I say, as a little monorail for the cells that have birth here to be able to go and know exactly where they need to go. Okay, so the cells in the mantle will eventually differentiate into neurons and glial cells. Neurons then will create synapses among themselves and send axons, as we can see, like in here. They send their axons. We're creating a cell pure of marginal zone. Okay, so here we have the marginal zone that we only see, what we see, just axons, right? So in here, in the mantle zone, because we have the bodies of all of the cells that have birth and stay in the mantle, this is the gray matter, because we have the bodies of the neurons, right? As they create the synapses and send it to the outer part of this neural tube, we only get the myelin axon, myelinated axons, excuse me. And that's what we call the white matter, because the white is because of the axons myelinated. Okay, so eventually glial cells cover most of the axons and the marginal zone with the myelin sheaths and produce the white appearance that give us this white matter. Now, the germinal epithelium of the ventricular zone, so the cells that are in this section, will later shrink and become the ependyma. So the ones that are going to create the cerebrospinal fluid. Okay. And lines will cover the cavity, the brain cavity. Is this clear up to here? Yeah, okay. Now, we're going to have a subsequent differentiation that depends on the positioning of these migrating neurons and which position they occupy once outside of the germinal epithelium. So we are going to talk about 3 different levels of organization of cells from the neural tube, depending on what level they are found. So we're going to talk about the spinal cord and medulla organization, the cerebellar organization, and the cerebral organization. So 3 different things depending on the level at which they are found. So I'm going to start with the spinal cord and medulla organization. Here we have asymmetric division, and pay attention when I say symmetric and asymmetric. Okay, we start with an asymmetric division that takes place in the ventricular zone. So once again, this is the basic architecture that we talked about 2 slides ago. So we start with the asymmetric that takes place in the ventricular zone. So this zone here that we say that is the ependymal zone, the ventricular zone. Okay, so this zone over here. The second part is the mantle or the intermediate zone that contains the body of the neurons. Okay. And then we have the marginal zone that contains the axons of the neurons that are found in the mantle. Okay. And we're going to have some connective tissue over there. So as this matures to this section, we see the appearance of the sulcus. So we are going from the very beginning. This is the presumptive ventral region. This is where the hinges, dorsolateral hinges, are forming for the fusion, elongation, and joining over here. Okay, and so this is the presumptive dorsal. As it gets elongated and the joining, we're going to find a point of differentiation between the dorsal and the ventral part, and that is what we call the sulcus limitem. Okay, so the sulcus limitem, I have something that I didn't click. The sulcus limitem that we have in here divides the dorsal from the ventral halves. Okay, so let's see what happens. Okay, come on, here we go. So when we do a cross-section of the spinal cord, this is what we have. So we have spinal nerve. This is our dorsal section. This is our ventral portion. Okay. So the dorsal portion receives inputs from sensory neurons only, while the ventral portion is involved in affecting motor functions. This is the same thing as we saw, we say on Tuesday, right? Dorsal is always sensitive, ventral is always motor. Sometimes we're going to find the interneurons, depending on at what level we're talking about on the spinal cord, interneurons in this section. But what you need to retain is dorsal portion of the spinal cord is always sensitive, and ventral portion is always motor. The second level that we wanted to talk is the cerebellar organization. So in the brain, cell migration, differential neurons, and proliferation depends on the position. And so the initial three-zone architecture now gonna be more complicated, more complex, okay? So the cerebellar development results in the formation of cortex, which is highly folded. All the convolutions in the brain are due to this structure of the outer portion. Okay, so the outer region, and this is what we have in here. So we have the three zones now become more zones in here. So in the cerebellum, some neuroprecursors enter the new marginal zone. Okay, so we have some of these cells get into this mantle zone, but they don't stay there necessarily. Okay, some gonna go far away to the outer, and this is the external granular layer. Okay, so and these are always to the outer boundary of the neural tube. The inner compartment, so we're gonna have some cells that are gonna start migrating now towards this section. Okay, so the inner compartment of the external granular layer contains neural progenitors, cells that start proliferating and get in contact with cells that secrete, here we go, BMPs. These BMP molecules that is telling them where to go and become the precursors of the major neurons of the cerebral cortex that are the granule neurons. So we come from this ventricular, but then we go to this external granular, and they start migrating in this direction under the influence of BMPs. So these granule cells migrates backwards towards the ventricular zone. So that's what I say, they are going now to the ventricular zone. Okay, and they will form this internal granular layer. Okay. Meanwhile, the original ependymal cells, the ones that we have in here, okay, gonna start creating some glial cells, including the Purkinje cells. So the granular layer rises from this external granular layer under the influence of BMPs. Meanwhile, the ventricular zone stem cells going to generate the Purkinje cell layer over here. Okay. This is a major type of cell that we all find in the cerebellum. So they play a very important role in here. That control, what do the Purkinje cells control in our body? Do you remember? No, the movements of our body is all under control of the Purkinje cells. Okay, so very important, and all of those comes from this ventricular zone over here. Now, Purkinje cells gonna make the secretion of one of our best friends in development, Sonic hedgehog, okay, which sustains the division of the granule neurons precursors in the external. So you see how these ones are so close to the granular external cells. So now, because the Purkinje cells place themselves just nearby them, by the secretion of Sonic hedgehog is going to maintain the proliferation of this external granular layer, so they can migrate to form this internal glandular layer. Okay, so each Purkinje neuron has an enormous dendritic arbor, which spreads like a tree, really wide and really bulky, like, okay. So a Purkinje cell neuron normally has as many as 100,000 synapses with other neurons, and is more than any other neuron in our body. The last level of organization of the neural tube is the cerebral organization. And in here, the three-zone arrangement of the neural tube is also modified in the cerebrum, and is called the cerebral cortex. Okay, so this is what we have in this zone over here. So the cerebrum is organized now both vertically and horizontally. It's not just horizontally. In this case, it's also vertically, and we're going to see that in one slide in a couple of minutes. Okay, so the progenitor cells migrate through the intermediate, so the mantle zone over here. Okay, on the processes of the radial glial cells. Okay, so migrate from here to from this cortical plate through some of these radial glial cells to generate the zone of neurons on the outer surface over here. Okay, and that is what, oops, I clicked too quick. Well, we're gonna have this cortical plate over here, and this is the new layer of the gray matter that we're gonna be calling the neocortex. Okay, a specification of this neocortex involves a transcription factor that is the LHX2 transcription factor, which activates many other cerebral genes. So far we have BMP, Sonic hedgehog, and now we have the LHX2. So all transcription factors are really important into this. So here is the neocortex stratification. This neocortex that started just like a simple layer, right, of cells. Now we're going to have 6 layers of neural cell bodies, and those are the ones that we have here, 1 to 6. Okay. So we have the full maturation of this layer takes many years to be achieved. In general, to middle of the childhood, whatever that means, depending on the kids. Okay. So mid-adolescence is still, you know, sometimes kind of like still developing in there, and we're gonna watch a video on that. So each layer of the neocortex differentiates from the others in its functional properties, the type of neurons that it has, as well as the set of connections they are capable to make. So because this highly hierarchy and functioning of the cerebral cortex is also organized horizontally into more than 40 regions that regulates anatomically and functionally different processes in our body. So we have 6 layers vertically and 40 layers horizontally, okay? So this is the molecular stem cells, and then we have the granule cells, or pyramidal cells, more granule cells, pyramidal cells, multiform. You see, so we have a huge variation. But we don't want to stop too much on this because you can do 1 semester just on brain anatomy and type of cells. And there is one inside, so we're not going to go there. So the vertebral brain, as I say, is highly complex and required to have all the right pieces of the puzzle in the right position, and each piece must be placed at the right time. It's not like when we build puzzles. Do you like to do puzzles? I normally start on the corners and then I start filling in, right? Some people start separating by colors, and then some people start just on the bottom and start building up. So there are different strategies. Well, that doesn't work in the brain. There is only one strategy, and it's having the right type of cell at the right time in the right position, okay? So for the brain, as any good household, is constructed from the bottom up, okay? So in the developing brain, stem cells and progenitor cells division is precisely controlled in order to have the right number and type of cells. So here we have, these are the embryo cells. Here we are just forming that brain, that tube, right? And here is the adult. So we see that we have a larger variety of cells in the adult than we have in the embryo, right? So in order to position these cells as they are being produced relies on a scaffolding made by our radial glial cells. These are the common elements between the two, okay? These radial glial cells are responsible for putting the right type of cell in the right position. So in this figure, we observe the regional specification of the embryo radial glial cells. Here, there were a couple of scientists, you can read that into your book. They discovered that differentiated neurons of the cortex were descended from stem cells that reside in comparable areas in the embryo, okay, which themselves normally come from the ventricular zone, so those stem cells, so the ones that are at the lumen, right? So we see that the ventricular radial glial in the embryonic brain, the ones that are here, you see the positioning of these ones. These are the purple, the green ones, and the blue ones, occupy more or less the same positions in the adult, okay? So in here we see that the ventricular radial glial normally, and the clonal derivation of the B type, this is the B type, occupy about the same position, and so on, with the green ones and the blue ones, okay? So we have different type of cells in here. We have the GC. GC is simply for granule cells, the ones that I said that occupy the bulk, okay? We also have some, excuse me, periglomerular cells. We have other molecules in here. So that doesn't matter too much to the point, but the big point is that the position is relatively the same. So this model demonstrates that the ventricular zone of the radial glial are regionally specified in the embryo and give rise to similar specified adult stem cells as they propagate. Here is when things start getting a little more complex. And this is just showing the neurogenesis, okay? So in here we have that stem cells can divide symmetrically to produce 2 copies of itself, thus creating the pool of stem cells. Okay, so one stem cells divide, one of those cells remain as a stem cell, and the other one will differentiate depending on the factor, right? On the transcription factor that is subjected to. But also we can have some asymmetrical divisions, okay? So this asymmetrical to self-renew and yield a differentiation daughter cells, okay? So, and in here, it's the beauty of nature. During neurogenesis, a pool of homogeneous stem cells first undergo proliferative expansion and diversification, and then initiates the production of successive waves of neurons. So in here we have some group of progenitor cell types: neuroepithelial cells, ventricular radial glial cell, intermediate progenitor, or a radial glial cell, okay? And we have the different type of cells that they differentiate: deep layer neuron, upper layer neuron, and microglia, either oligodendrocyte or astrocytes. So as the neural tube matures, the progeny of the neuroepithelial stem cells become radial glial cells. So we are here right at the bottom. This is the pre-neurogenesis, and this is late neurogenesis, okay? So we start with a neuroepithelial cell that goes mitosis, and at the end, we have two. One will remain as a stem cell. So this is the symmetrical proliferative section that I say that can produce two differentiated daughter cells, and one will remain cell, okay? So this division of radial glial cells occurs always at the level of the lumen, right? So in this ependymal section, closer to the lumen of it, the central canal, okay? In the cerebrum, as the progenitor cells delaminate from the ventricular, so we are moving to this end, okay? They will form the subventricular zone. So they are moving from this zone to this zone. So we are here, and we're going to go to the subventricular zone that is going to be over this section, okay? And all of this, if you look at it, they are all still attached to the base. That's why we say from the bottom up, okay? So a single stem cell in the ventricular layer can give rise to both neurons, the glial cells and any of the cortical layers which we described in a couple of slides ago, okay? So in this image, we see three major progenitor cells in the germinal strata. So we have the, where is it here? So this is the transition, initial transition. This is the neuroepithelial cells. Gonna go through a transition to the ventricular radial glial cell, so the VRG. But also, depending on what type of element is stimulating them, they can take different fates. And this is the asymmetrical neurogenic transition, because depending on the factor, either they become a neuron, as we have in here, or they remain as a neuroepithelial cell, okay? So we also have the outer radial glial, that is the ones that have, I think, here we go. So this is the intermediate progenitors, continue with an asymmetrical neurogenesis, but in this case we have the ORG, this is the outer radial glial cells. And we see how they still divided in an asymmetry, right? Or we have the intermediate progenitor. The intermediate progenitor is the IP over here. So through the phase of brain development, the VRGs, the ventricular radial glial cells, serve as the parental stem cell type And in addition to directing generating neurons, it also gives rise to both the ORG and the IP cells. Okay, so just this one that started here can give rise to 2 and even 3 different types of cells. Is that clear? Okay, good. So in the end, self-renewing symmetrical division dominate early in neurogenesis. So this new part over here of neurogenesis, to expand the progenitor pool, and then more asymmetrical divisions take place for progenitor cell differentiation. Okay, so now we talk about this glial guidance, how these cells know where to go and how, right? So the development of the spatial organization, either whether it is vertical or horizontally, is very critical for the proper functioning of the cerebellum, for instance. Okay, so one mechanism that is important for positioning young neurons in the brain, it is the glial guidance. So through the cortex, neurons ride a monorail within these glial cells. Okay, and if you were in class, you will be seeing me just flapping my arms just to illustrate this. Okay, so they take a specific, this is the monorail, this is the leading process, this is the glials, this is the neuron. So it just wraps around and knows exactly. And this is a microphotography that was taken through time. And you see how that cell is moving, it's advancing when it attaches to this monorail. Okay. So granule cell precursors travel along the processes of the Bergman glial cells, and the neurons maintain this addition to the monorail using some proteins that we call astrotactin. So astrotactin is the molecule that helps them to glue to that monorail and don't lose contact until they get where they need to go. Okay, so astrotactin addition protein is needed by the granule neuron, cell neuron, to maintain their addition to this glial process. And then the direction appears to be regulated by a complex of different events of the brain. And we're going to have the BDNFs. This is brain-derived neurotrophic factors. Okay, that is a paracrine factor made by the internal granular layer. And that's how they attract to one another. So the granular layer creates the BDNFs, and this is specific for the astrotactin, and that's how they maintain that addition. So after the initial mitosis that occurs in the ventricular portion, right? Most of the neural precursors generated in the ventricular ependymal zone are migrating outwards. So this is what we have here. Okay, and they do it using the glial process. Okay, until they go into the cortical layer or plate, which is the outer surface of the brain. Okay, so as in the rest of the brain, those neural precursors with the earliest birthdays from the layer closest to the ventricle. So it's like when you try to go sometimes into a bus or into a metro. I don't know why some people like to be front, and then you need to open your way to the back that is empty. Okay, so this is what they do. The cells are birthed early, they're going to remain closest to the ventricular zone. The cells that birth later, they need to move to the back, to the different zones at the back. Okay, so in here we have some cells. This zone was occupied, and then these are moving in. So in this figure A, what we have is the early birthdays cells. So here we have at the initial, so we have the percent of twin labeled neurons, so we know more or less how many neurons we have and each of the sections. So you see 6 is the closest to the ventricular zone, right? So this is what we have a little bit more over here, okay? In B, we have the late birthday cells. So 6 is occupied and they're going more towards the back of that layer, so more toward the outside of the cortex in there. So second neurons must travel larger distances to form the more superficial layers of the cortex. So inside-out gradient development from inside to the outside gradient. Here in figure C, we observe that if the early precursors, which is the dark blue here, are transplanted into older ventricular zone after the last mitotic S phase, okay, the neurons they form migrate to layer 6, indicating that those cells were committed. It's just because we took them a little late, okay. In D, we have that if these precursors are transplanted before or during the late S phase, they migrate with the host neurons in layer 2, meaning that they are feeling that they just were born, okay? So remember, early born neurons establish the deeper layers and later born neurons from the more superficial layers, okay? So, which means that the cerebrum is growing from inside to the outside. Okay, so as cells go through the cell cycle, the nucleus moves within the cells, and this is what we see in this image. This is the asymmetrical division of these radial glial cells. So when the nucleus is in the S phase, do you remember what happens in the S phase? Because I have mentioned it like 3 times now. Do you remember what happens in the cell division phase S? Is the DNA synthesis, okay? The nucleus is near the exterior edge. So here we have the lumen. This is the exterior of the two, okay? So this is the ventricular zone closest to the lumen, and this is a cut like this, okay? So this is the S phase. We see that the nucleus is more towards the outside of the tube, right? As the cell cycle proceeds, the nucleus moves towards the lumen, okay? So to the interior of the tube. And mitosis occurs when the cell is closest to the lumen, not before. Okay, so here we go. This is the S. They start transforming, condensing the chromosomes, and whoops! So now that is the closest to the lumen. There is metaphase happening in here. Okay. So the division of the neuroprogenitor cells at the lumen regulates the exposure of the cells to different paracrine factors that we see here in this little box, okay? So all is expressed. So if we look at the outer side, we have a lot of encaderins, okay? In the ventricular zone, once the closest, we see a high proportion of part 3. Okay. And as they divide, still at the lumen, one of the cells we can see here gonna have high concentration of notch, and the daughter cells have low concentrations of notch. Okay. So in here we have 2 differential migration patterns of the cells. Cells from the outer cortex of an adult brain, they came from the germinal neuroepithelium, okay? As in the figure, there are 2 waves of cell migration, the symmetrical, horizontal, okay, division that produces two cells that are attached. So this is horizontal, right? Produces two cells that are attached to the lumen. And this is in anaphase to telophase, okay? And then this symmetrical division is changed to an asymmetrical division, which is where one of the cells remain attached to the lumen. This is the one that has high concentrations of notch, okay? And this one continues to be a stem cell, okay? But the daughter cells will detach because it has low concentrations of notch and detaches from the lumen and migrates in the differentiated into the glioblast somewhere, okay? So the migration of cells is regulated by PAR proteins. You see that is what is normally specifying here. So PAR is responsible of the migration of the cells and the cerebrospinal fluid. So during mitosis, this PAR that is illustrated with the green color becomes concentrated in one of the daughter cells. And this is what we see it a little more in this one. This one, the daughter cells are gonna go into the asymmetric movement, has encadering here, okay? So the expression of NOT causes the cells to remain a stem cell and no longer divide at that point. And then we can move back again into this. Is this section clear about this symmetric horizontal migration and asymmetric detachment, all regulated either by PAR or notch? Okay. So one interesting thing with, with brain development is that even though we are all vertebrates, it's not equal among the different species. So development of the human neocortex is particularly striking. And this is what we believe, or we like to believe, when we are more evolved species, okay? So the human brain continues to develop and proliferate at a fetal rate even after birth. This is one of my first assignments today. So for our size, based on comparison with other mammals, human gestational period should be about 21 months. Imagine that. I kept my babies for 9 months and I had enough after the 9 months. Okay, but if we did, the head would be too large to go through the birth canal, so no happening. So it has been suggested, as I say, that we are extrauterine fetuses because normally the brain continues to grow during the first year. By using some electroencephalograms of brain activity, the first sign of brain activity normally is seen at 7 months of gestation. Okay. At 12 weeks, the cerebrum has no neural connection and has no thought, no conscientiousness, or not fear at all. So, and I may be putting my feet here in a booby trap. We define death. How would we define death of an individual? Not all at the same time, please.
From the ethics class that I took last summer, there are 2 or 3 stages at which death can be declared. When it is on the surgical table, it is either brain dead or when organs are shutting down. When it is a gradual decline of the person, I believe they are declared dead when they are brain dead.
Brain dead, exactly. So when we don't have any signal from the brain, we normally declare the person to be dead, right? But perhaps the acquisition of the brain activity could be used as a definition of the start of human life, right? So with these advocacy groups of abortion or against abortion, what is alive? Yeah, but doesn't have any brain activity. Okay, I am not taking one side or the other one. That this is based on personal conscientious feeling, right? How you feel about it. But that is another option. If we define death as not brain activity, why we can't define to be human life definition, right? When this individual gains some electro brain activity. So after this complex series of events that are required for the nervous system formation, it is clear that this sophisticated organ is far from being simple. It requires so many things to be placed in the right position. It is proposed that the number and intricacy of the jury, so the convolutions, how many of these convolutions here we have, right, of the jury, and the sulci present, so this section over here, in the brain of vertebrates is directly correlated with learning capabilities. So this is what we observe in this figure. We observe the cerebral cortex of a human, which is considered gyroencephalic because it has a lot of gyri, right? Elephants also have a very same complex cortex as humans, and we know that elephants are highly hierarchical and highly intelligent, okay? In the figure below, we see the cerebral cortex of a mouse, which is considered disencephalic, lisse, smooth, okay? So it have a very few gyri and almost no folds at all, okay? So the amount of gyrification is always correlated with intelligence. For the longest time, we thought that the brain growth undergoes a rapid initial growth of neurons during the fetal and early childhood, and then it will stop growing. However, there is new evidence that shows that the brain continues to grow until late adolescence puberty, and that the different areas of the brain have different rates of maturity. It's not equal. If we compare the brains of a boy, of a girl, there are also some differences in there in the level of at the time at which they reach a maturity. We know that after puberty, brain growth start going down. They start pruning, so eliminating some connections that they are not needed anymore. Okay, that were important at the beginning, but they are not anymore. And there is also a direct link between pruning and language acquisition. So difficulty would explain clearly why kids learn language a little more easy than adults. If you were a kid and you were raised in a bilingual environment, you use both languages with no difficulty whatsoever, not even the accent, right? When we start learning a language, when we are a little older, it starts becoming a little harder. So this is what I have. So my accent that I have is part of my identity. That's the last thing that I want to lose, right? But also because my tongue knows how to move to pronounce certain words, and that sound doesn't exist in my ear yet for another language. So I know 4 different languages, and sometimes I'm thinking in one and then I speak and my mouth gets in another one. So it starts getting a little more complicated. So in here, we have a brain activity, brain cortex, showing this is the primary motor cortex and this is the sensory one. This is age in years 5. This is 8. This is 12, 16, and 20. And if we look at the degree of myelinization, so the percent of white matter, look at this at 20, we're just getting all those connections a little better, right? So myelinization is essential for the rapid transmission of information and proper neural functioning. And it's high when people are around in the 20s, okay? I would say that here early, it doesn't seem to be that much. And here we have this, the teenage brain. So they are prone to higher risk behavior because they are still need to learn. So the prefrontal cortex, that is that section that allows us to plan, to reason, grows until 25. So this is why some of you are still doing the same mistakes every time. I didn't learn, right? So we are measuring what are the consequences if I do this. And this says it reaches normally at age of 25. Sometimes it's a little farther than that. We have the amygdala here. That is the emotional, so we can correlate situations and emotions in a little different way. The way how we reacted when we took tantrums when we were small, they're not necessarily now present. So having said that, I just want to show a video that discusses this. It's coming. It's coming. Come on. There we go.
15 years ago, it was widely assumed that the vast majority of brain development takes place in the first few years of life. Back then, 15 years ago, we didn't have the ability to look inside the living human brain and track development across the lifespan. In the past decade or so, mainly due to advances in brain imaging technology such as magnetic resonance imaging or MRI, neuroscientists have started to look inside the living human brain of all ages and to track changes in brain structure and brain function. So we use structural MRI, if you like, to take a snapshot, a photograph at really high resolution of the inside of the living human brain. And we can ask questions like, how much grey matter does the brain contain, and how does that change with age? And we also use functional MRI, called fMRI, to take a video, a movie, of brain activity when participants are taking part in some kind of task, like thinking or feeling or perceiving something. So many labs around the world are involved in this kind of research, and we now have a really rich and detailed picture of how the living human brain develops. And this picture has radically changed the way we think about human brain development by revealing that it's not all over in early childhood, and instead, the brain continues to develop right throughout adolescence and into the 20s and 30s. So adolescence is defined as the period of life that starts with the biological, hormonal, physical changes of puberty and ends at the age at which an individual attains a stable, independent role in society. It can go on a long time. One of the brain regions that changes most dramatically during adolescence is called prefrontal cortex. So this is a model of the human brain, and this is prefrontal cortex right at the front. Prefrontal cortex is an interesting brain area. It's proportionally much bigger in humans than in any other species, and it's involved in a whole range of high-level cognitive functions, things like decision-making, planning what you're going to do tomorrow or next week or next year, inhibiting inappropriate behavior, so stopping yourself saying something really rude or doing something really stupid. It's also involved in social interaction, understanding other people, and self-awareness. So MRI studies looking at the development of this region have shown that it really undergoes dramatic development during the period of adolescence. So if you look at grey matter volume, for example, grey matter volume across age from age 4 to 22 years increases during childhood, which is what you can see on this graph. It peaks in early adolescence. The arrows indicate peak grey matter volume in prefrontal cortex. You can see that that peak happens a couple of years later in boys relative to girls, and that's probably because boys go through puberty a couple of years later than girls on average. And then during adolescence, there's a significant decline in grey matter volume in prefrontal cortex. Now that might sound bad, but actually this is a really important developmental process because grey matter contains cell bodies and connections between cells, the synapses. And this decline in grey matter volume during prefrontal cortex is thought to correspond to synaptic pruning, the elimination of unwanted synapses. This is a really important process. It's partly dependent on the environment that the animal or or the human is in that synapses that are being used are strengthened, and synapses that aren't being used in that particular environment are pruned away. You can think of it a bit like pruning a rose bush. You prune away the weaker branches so that the remaining important branches can grow stronger. And this process, which effectively fine-tunes brain tissue according to the species-specific environment, is happening in prefrontal cortex and in other brain regions during the period of human adolescence. So a second line of inquiry that we use to track changes in the adolescent brain is using functional MRI to look at changes in brain activity across age. So I'll just give you an example from my lab. So in my lab, we're interested in the social brain. That is, the network of brain regions that we use to understand other people and to interact with other people. So I like to show a photograph of a soccer game to illustrate two aspects of how your social brains work. So this is a soccer game. Michael Owen has just missed a goal, and he's lying on the ground. And the first aspect of the social brain that this picture really nicely illustrates is how automatic and instinctive social emotional responses are. So within a split second of Michael Owen missing this goal, everyone is doing the same thing with their arms and the same thing with their face. Even Michael Owen, as he slides along the grass, is doing the same thing with his arms, and presumably has a similar facial expression. And the only people who don't are the guys in yellow at the back. And I think they're on the wrong end of the stadium, and they're doing another social emotional response that we all instantly recognize. And that's the second aspect of the social brain that this picture really nicely illustrates, how good we are at reading other people's behavior, their actions, their gestures, their facial expressions, in terms of their underlying emotions and mental states. So you don't have to ask any of these guys, you have a pretty good idea of what they're feeling and thinking at this precise moment in time. So that's what we're interested in looking at in my lab. So in my lab, we bring adolescents and adults into the lab to have a brain scan. We give them some kind of task that involves thinking about other people, their minds, their mental states, their emotions. And one of the findings that we've found several times now, as have other labs around the world, is part of the prefrontal cortex, called medial prefrontal cortex, which is shown in blue on the slide. And it's right in the middle of prefrontal cortex, in the midline of your head. This region is more active in adolescents when they make these social decisions and think about other people than it is in adults. And this is actually a meta-analysis of 9 different studies in this area from labs around the world, and they all show the same thing, that activity in this medial prefrontal cortex area decreases during the period of adolescence. And we think that might be because adolescents and adults use a different mental approach, a different cognitive strategy to make social decisions. And one way of looking at that is to do behavioral studies, whereby we bring people into the lab and we give them some kind of behavioral task. And I'll just give you another example of the kind of task that we use in my lab. So imagine that you're the participant in one of our experiments. You come into the lab, you see this computerized task. In this task, you see a set of shelves. Now, there are objects on these shelves, on some of them, and you'll notice there's a guy standing behind the set of shelves, and there are some objects that he can't see. They're occluded from his point of view with a kind of grey piece of wood. This is the same set of shelves from his point of view. Notice that there are only some objects that he can see, whereas there are many more objects that you can see. Now, your task is to move objects around. The director, standing behind the set of shelves, is going to direct you to move objects around. But remember, he's not going to ask you to move objects that he can't see. This introduces a really interesting condition whereby there's a kind of conflict between your perspective and the director's perspective. So imagine he tells you to move the top truck left. There are 3 trucks there. You're going to instinctively go for the white truck because that's the top truck from your perspective. But then you have to remember, oh, he can't see that truck, so he must mean me to move the blue truck, which is the top truck from his perspective. Now, believe it or not, normal, healthy, intelligent adults like you make errors about 50% of the time on that kind of trial. They move the white truck instead of the blue truck. So we give this kind of task to adolescents and adults, and we also have a control condition where there's no director, and instead we give people a rule. We tell them, okay, we're going to do exactly the same thing, but this time there's no director. Instead, you've got to ignore objects with a dark grey background. You'll see that this is exactly the same condition, only in the no director condition, they just have to remember to apply this somewhat arbitrary rule, whereas in the director condition, they have to remember to take into account the director's perspective in order to guide their ongoing behavior. Okay, so if I just show you the percentage errors in a large developmental study we did, this is in a study ranging from age 7 to adulthood, and what you're going to see is the percentage errors in the adult group in both conditions. So the grey is the director condition, and you see that our intelligent adults are making errors about 50% of the time, whereas they make far fewer errors when there's no director present, when they just have to remember that rule of ignoring the grey background. Developmentally, these two conditions develop in exactly the same way between late childhood and mid-adolescence. There is an improvement, in other words, a reduction of errors in both of these trials, in both of these conditions. But it's when you compare the last two groups, the mid-adolescent group and the adult group, where things get really interesting, because there is no continued improvement in the no director condition. In other words, everything you need to do in order to remember the rule and apply it seems to be fully developed by mid-adolescence. Whereas in contrast, if you look at the last two grey bars, there's still a significant improvement in the director condition between mid-adolescence and adulthood. And what this means is that the ability to take into account someone else's perspective in order to guide ongoing behavior, which is something, by the way, that we do in everyday life all the time, is still developing in mid to late adolescence. So if you...
Okay, it is 11:22. I just want to add that link into the news. If you want to be able to see the rest of the video, you can see it, but I just want to give you the opportunity to finish that. If you need to go, then you need to go, right? So just remind you that tomorrow we will have a lecture in the lab, so we can advance a little bit more on the lecture side, so we can finish also the semester a little earlier. I will be posting the first study case today, later today, okay? And I already have the term paper organized, but we want to talk about the term paper tomorrow, okay? I'm gonna make a little pool to see. I think that you can all tell me, but I'm gonna put the time for the term paper to be submitted, not at the end of the semester as is indicated in the syllabus. I think I have said the 2nd of April, but I'm gonna push it into the 15th of April. That is normally the date at which we should have an exam. And that's what I wanted to have that date. As remember, we don't have an exam, it's a term paper. Okay, but I wanted that date to see up to when I will be able to extend that. So I think that is a good thing to have a little bit more of time. I will be submitting, uploading that document tomorrow, but we can talk a little bit more tomorrow. Catherine, I know that you won't be there tomorrow, but if you agree that on the 15th it's a good date. I saw you doing yes, so I take that as a yes. But we're gonna do it. Doesn't mean that you cannot submit by the end of April. It all depends how organized you are, okay? I just wanted to give a little bit more of time, although I know that sometimes when I give more time, it means that you say, oh, I have more time, I'm gonna do it just on the last week. And you don't need to be catastrophic if you just leave it for the last week, because you need to do a live review search. So you're gonna need to look and read papers and a lot of things like that. I'm giving you like 5-6 different videos, and each video is like an hour to watch. So that's a lot of work, but remember, it's 25 of your final mark is that assignment. So that many hours gonna need to be invested. So having said that, I see you tomorrow at the time of the lab, and we are going to lecture, okay? If we submit at the end of April, the deadline is the 15th. So there is no more. April 15th is the last day that you can submit, and it will be at noon, okay? So just for the person, I don't know who asked that question, but I mean, you're gonna have a month and a half. So the sooner you start, the quicker you're gonna be able to do it, okay? after that date, there is no acceptance. Okay. Okay. Talk to you a little bit more about that tomorrow. Bye-bye.